Welcome to this explainer. Today, we're stepping into the really fascinating world of microscopic detective work. We're going to explore exactly how scientists and medical pros track down those tiny suspects to diagnose bacterial infections. So grab your lab coats because we're jumping right into the nuts and bolts of specimen selection and direct microscopy.
Seriously though, have you ever wondered how we actually see the invisible culprits making us sick? Picture this. A patient comes in with a severe infection, and the clock is ticking. We literally can't afford to wait days for a time-consuming lab culture to finish before making a life-saving call. We need clues, and we need them fast. That is exactly where the incredible tool of direct microscopy swoops in to bridge that gap. So, here is our roadmap for today's investigation. First, the first diagnostic step. Number 2, selecting the right specimen. 3, preparing the sample. 4, the art of staining. 5, identifying the culprits. And finally, number 6, safety and limitations. All right, section 1, the first diagnostic step, the front line of early decision-making. Okay, let's dive right into this crucial first step. Direct microscopy is our rapid, super cost-effective frontline tool. Now, while it's not the final definitive answer, it's an absolute game changer for early decision-making. By simply looking under the lens, we can detect if bacteria are actually present, what general type they are, their shape, and exactly how they arrange themselves. Plus, we get to see the host's cellular response, things like pus cells or red blood cells actively fighting off the infection. This rapid intel guides our subsequent cultures and points us toward the right treatments way earlier. Moving on to section 2, selecting the right specimen, gathering the evidence. Now, what's really fascinating here is that a microscopic investigation is literally only as good as the physical sample you collect. Different conditions demand highly specific evidence. For instance, suspecting septicemia, you need a blood smear, though keep in mind, it has pretty low sensitivity. For mastitis, milk is our go-to, and we might even centrifuge it to concentrate the bacteria. Intestinal or enteric infections, we need a fresh feces sample. But here's a massive warning. In abortion cases, we use placenta or fetal tissues, but there is a severe zoonotic risk, meaning these diseases can jump straight from animals to humans. Handling these tissues carries incredibly high stakes. Oh, and for a urinary infection, an early morning urine sample is your best bet. Basically, the evidence has to perfectly match the crime scene. But it isn't just about what we collect, it's the strict rules of how we collect it. To be a viable specimen, it has to come directly from the active disease site and be completely free from any environmental contamination. We need a good quantity, and it must be properly preserved and labeled. But here is the most critical rule. Seriously, absolutely crucial. You have to collect the sample before any antibiotics are given. If a patient is already flooded with antibiotics, the bacteria might be dead or structurally warped, completely ruining our chances of identifying the viable culprits. That brings us to section 3, preparing the sample, prepping the canvas. So we've got our evidence securely in hand, now we have to physically prep it. We essentially have two main ways to turn that raw evidence into a viewable microscopic canvas. First, the wet mount examination. Here you just place a drop of fresh specimen directly on a slide, slap a cover slip on it, and examine it under low and high power. This is awesome for observing things in their natural fluid state. It lets us differentiate true microbial motility, like actual bacteria swimming around, from random passive bouncing, which we call Brownian movement. It's also great for spotting pus cells and spirochetes. On the flip side, we have the stained smear examination. This requires physical spreading, drying, and fixing of the specimen so we can use direct staining techniques. So how do we actually prepare that stain smear? Well, it's a very precise physical process. Step 1. Start with a rigorously clean glass slide. Step 2. Place a small amount of the specimen right on there. Step 3. Spread it evenly to make a thin smear. Step 4. Let it air dry completely. And step 5. Heat fix it gently, usually by just passing it quickly over a flame so the bacteria actually stick to the glass. Quick word of warning though, making a smear too thick is a total rookie mistake. Thick smears lead to terrible staining where everything just looks like a dark, unreadable blob. Thin and even is for sure the key to success here. Section 4, the art of staining, painting the picture. Let's get into the staining phase and see how this really builds our case by bringing invisible bacteria to colorful life. We've got two primary painting techniques. Simple staining uses a single dye, like methylene blue or crystal violet. It's fast and does a great job highlighting the basic shape, size, and arrangement of our suspects. But gram staining?
That is our masterstroke. It fundamentally differentiates bacteria into two distinct camps, gram-positive and gram-negative. It distinguishes them based on the complex architecture of their cell wall. So this isn't just about making them visible. It's about unlocking deeper structural clues that tell doctors how a bacterium behaves and ultimately how to treat it.
All right, Section 5. Identifying the culprits, reading the clues. Think of this phase as our detective's decoding manual, where we finally translate those colorful visual clues into a suspect's precise identity. When we peer through that microscope eyepiece, what are we actually hunting for? Well, we break it down into several features. For shape, are we seeing perfectly round cocci, rod-shaped bacilli, intermediate coccobacilli, or maybe twisted spiral forms? For arrangement, are they grouped in long chains, bunched up in clusters, or just sitting in neat little pairs? Then there's the staining result itself. Gram-positive shows up as a vibrant purple, while gram-negative appears distinctly pink. We also check the background for pus or epithelial cells, which tells us about the host's reaction. And finally, we hunt for special defensive forms, like tough spores or protective capsules. These specific combinations act just like puzzle pieces falling perfectly into place. For instance, if you see gram-positive, again, that's purple, cocci arranged in clusters, your clues are strongly pointing to Staphylococcus. But what if those same purple cocci are in chains? That points to Streptococcus. Spot some gram-negative pink rods, you might be dealing with Enterobacteriaceae. Pleomorphic rods, meaning they kind of vary in size and shape, suggest Pasteurella. And if you see those tough little spores, you're likely looking at Bacillus or Clostridium. It's literally a microscopic matching game with the highest stakes.
Finally, Section 6, Safety and Limitations, Knowing Our Boundaries. As much as we absolutely love this microscopic sleuthing, we've got to acknowledge that while it is incredibly powerful, it's not invincible. We have to respect its limitations. First off, we can't identify the exact specific species of bacteria just by looking at them. We can only group them generally. We also face low sensitivity in certain samples, like blood smears. In those cases, finding the culprit is honestly like finding a microscopic needle in a haystack. Plus, dead or non-viable organisms might be missed entirely or stain improperly. And complex scenarios, like mixed infections with multiple types of bacteria, those can easily confuse our preliminary results and make the slide look incredibly chaotic. And because we're dealing with live, potentially dangerous pathogens, biosafety is absolutely non-negotiable. You've got to wear gloves and a lab coat. You absolutely must avoid aerosol formation, meaning splashing or spraying the sample. Creating an invisible mist of bacteria is pretty much the fastest way for the investigator to get infected by the exact culprits they're hunting. Not good. Additionally, we have to flame our slides carefully, thoroughly disinfect the microscope stage after we're done, and strictly follow proper waste disposal protocols. Safety first, every single time. Which brings us to a quote that perfectly illustrates the golden rule of our entire journey today. Microscopy suggests identity, but culture confirms it. Direct microscopy gets us on the board quickly. It points us in the right direction and lets doctors start critical treatments fast. But we must always rely on the subsequent laboratory culture to actually grow the bacteria and lock in that final, definitive diagnostic answer. We've really just scratched the surface of diagnosing a bacterial infection using nothing more than a glass slide, a drop of stain, and a lens. It's pretty amazing that a single drop of fluid holds that much life-saving information. Which leaves us with this final thought. What other invisible worlds are waiting to be uncovered right in front of you? Keep observing, stay focused, and never stop questioning what you can't see with the naked eye. Thanks for joining me on this explainer.